Depression, insomnia, and cognitive decline do not exist in isolation. If you suffer from one, you are highly likely to experience the others. Biologically, they are deeply intertwined, with dysfunction in one area of the brain quickly spilling over into the next. Conventional medicine usually isolates these symptoms. A diagnosis of depression leads to an SSRI for serotonin, while sleeplessness is met with a targeted sedative. This approach treats specific neural pathways as if disconnected from the rest of the network. Long before we isolated specific neurotransmitters, traditional medicine systems employed a different strategy. Historical texts detail the use of Crocus sativus, the plant that produces saffron, specifically to calm the mind and treat melancholy. Modern clinical data shows these ancient applications are grounded in biochemistry. Standardized saffron extracts perform toe-to-toe with gold standard synthetic drugs, matching the efficacy weights of fluoxetine for depression and Donepel for slowing the symptoms of Alzheimer's. While those mood and cognition scores improved, patients also reported significant increases in restorative sleep and a corresponding drop in insomnia severity. This raises a crucial biological question. We need to understand how a simple flower stigma, the world's most expensive culinary spice, manages to rival highly engineered pharmaceuticals across 3 distinct brain disorders at once. The answer lies within the plant's chemical architecture. These flowers produce a profile of over 150 distinct bioactive compounds that function as a coordinated network. The first major player in this cluster is saffronol. This is the volatile oil responsible for saffron's aroma, and it operates directly at the neural synapse to regulate mental health. When neurotransmitters like dopamine and serotonin are released, specific transport proteins normally sweep them away. Saffronol acts as a competitive inhibitor, physically blocking those transporter gates. By obstructing these clearance pathways, critical chemicals pool and remain active much longer. This targeted inhibition allows saffron to alleviate clinical depression by keeping the brain bathed in its own neurotransmitters, achieving the biological results of an SSRI through a botanical pathway. This mood-stabilizing effect runs parallel to a physical response in the HPA axis, the biological loop connecting the hypothalamus, pituitary, and adrenal glands. When you encounter a stressor, this axis floods the bloodstream with cortisol. Saffron blunts this hyperactivity during moments of tension, where bioactive compounds in the extract actively suppress the secretion of that excess cortisol. We can map this effect through heart rate variability. In a stress test, the placebo group's variability plummets, signaling a nervous system in panic. But the saffron-treated group remains steady, buffering the cardiovascular system against the stress response. Lowered cortisol and a stable heart rate are the physiological prerequisites for sleep. Without excess adrenaline keeping the autonomic nervous system on high alert, the brain can transition into deep, restorative non-REM sleep. By addressing the biological stress response at the hormonal level, saffron repairs broken sleep architecture, which provides the biological foundation for a stable mood. Saffron's third major target operates through the water-soluble pigment that gives the spice its golden color, crocin. This compound operates via the absorption paradox. In native form, the crocin molecule is too large to cross the stomach lining. Bypassing the stomach, it enters the colon intact, where specific gut bacteria metabolize it, cleaving its sugars to transform it into smaller, highly absorbable crocitin. This bacterial processing allows the resulting crocitin to enter the bloodstream and readily cross the highly selective and restrictive blood-brain barrier. Inside the brain, crocitin stimulates the production of brain-derived neurotrophic factor, or BDNF. This protein triggers the growth of new synapses and helps clear out the amyloid beta plaques associated with cognitive decline. Saffron's neuroprotective properties depend entirely on this interaction with the microbiome. Without these specific bacteria, the extract cannot activate the pathways required to protect the brain from structural atrophy. Despite this biological potential, simply eating saffron in culinary dishes is not a reliable treatment for clinical depression or cognitive decline. The harvest is incredibly labor-intensive. It takes approximately 150,000 hand-picked flowers to yield a single kilogram of the spice, meaning raw plant matter varies significantly in potency. Rigorous clinical trials depend entirely on standardized extracts. These processes guarantee a precise, concentrated fingerprint of bioactive compounds, ensuring the brain receives the exact molecular dose required for a therapeutic effect. Saffron is effective because it utilizes network pharmacology, a biological system that simultaneously targets inflammation, neurotransmitter levels, and the gut-brain axis. Because sleep, mood, and cognition are biologically linked, a failure in one system destabilizes the others. A multi-targeted problem requires a multi-targeted solution. By bridging ancient usage with evidence-based psychiatry, Crocus sativus demonstrates the efficacy of treating mental health through complex, multi-targeted synergy.